As Suzanne mentioned, our paired in 101 cycle run may take as many as 10 days. So it's really important that we follow the progress of the run. We'd like to know that all the chemistry is occurring correctly, and we can do that by looking at our high-seq control software. On this, what we have over here is actually where the camera is imaging on the flow cell. The flow cell has approximately 8 lanes, and it's going to image one lane at a time. Each lane is divided into different swaths, and in addition to that, it can also image the bottom and the top of the lane of the flow cell. This little ball here represents where the camera is moving along the lane, and in a few minutes here, what you see is all of the fluorescence intensity together in the image of where the camera is progressing along the edge of the lane. These are all of the individual clusters that Suzanne mentioned are formed in our CBOT. During this particular cycle, one base has been added. Remember, each base has a different fluorescent tag. At the end of that, because only one base has been added at a time, at the end of the imaging cycle, all of those bases will be, all of those fluorescent tags will be removed, and we are able to start then the next cycle of sequencing. Again, another base will be added, so each one is done in succession, one base at a time. We image one cycle at a time. The other thing I'd like to show you here is another way that we monitor how the run is progressing. And in this, we're actually, the software, as we're cycling through these different sequences, the software is managing to look at what we are calling quality scores. And we'd like to know that during the run that we're getting good quality scores, meaning that each cluster is actually being able to determine one base at a time. So you can imagine if each cluster has got approximately 1,000 different templates, we would like them all to be C at one time or G at one time, but it may not be accurately that way. So the software does what's called a quality scoring, and so it is able to look at all of the fluorescence of that one cluster and say, yes, most of this would represent G or most of this would represent C. So we can see that over in this far graph here. Right now we're looking at lane 8, we're looking at both sequences, and we can see that, and quality scores are given as Q scores, and we can see that for what we consider to be a very high quality, that approximately 91.6% of all of those clusters have a quality score that is very acceptable for us. Down here in this lane, we're actually looking at cluster density across all 8 lanes. The blue boxes represent the total number of clusters, along with plus or minus in an average, from lane 1 all the way to lane 8. What you see on the green boxes are what the clusters that we consider to pass filter. So the software is also able to tell us the quality of the clusters as well. And those that pass filter are basically, we're being able to say this cluster is different from this cluster. And we want these boxes to match up or be on top of one another as much as possible. And as you can see for this run, we're very happy with our pass filter percentage as well as our quality scores. In this box here, what we're looking at is the intensity of each of the bases. So our red line is the intensity of A's across all these cycles from 0 to, or actually from cycle 1 to cycle 100. Our lowest one down here is G, and that's because G has the furthest distance to go in terms of being able to measure the fluorescence intensity. Our boxes over here, or actually our lanes, are looking at the cluster density on the lane, and we can also look at signal to noise. Here this is looking at the intensity, and in this case it's just the top surface, both surfaces. These metrics I can actually look at by remote software, so even on the weekend when I'm at home I can know how well this run is going. At this point, now that we've got all these cycles, we've got lots of sequences coming out, and Suzanne's going to describe where these sequences go.
So we've now reviewed the steps that are going on in terms of building the DNA itself and generating the sequence image. But you're probably wondering where this information is going, and if you are, that's a very good question. We're generating terabytes of information over a very short period of time. It used to be the case that we collected the image files and saved them, but sequence is now being collected so rapidly that's no longer feasible. So actually we collect some images, and in real time those images are converted over into DNA sequence material. We do that in collaboration with the Institute for Genomics and Bioinformatics here at UCI. So the information is actually first collected as an image file on the server in the facility, then it's piped across the campus 10 gigabyte intranet to the Institute for Genomics and Bioinformatics. There, an ELAN pipeline, a proprietary Illumina pipeline, is used to generate the actual base calls. At that point, there are several options for our users. They can receive either a FASTQ file downloaded to them, or they can choose to have the informatics staff align the sequence to what we call a reference genome. In the case of a human genome, for example, it would be a human reference genome. And then they could assemble the specific human sequence in which they are interested. So that concludes basically the information that we'll be giving you today about sequencing. Keep in mind that this human DNA sequence now is what is going to empower personalized genomic medicine in the future. So it's the ability to know the individual sequence variation among even basically normal human beings that will enable us to tailor pharmacologic treatments or to make predictions about potential predispositions to diseases in the population. So I hope you found our tour of the facility interesting. If you would like more information about the facility, that information is available on our website, and we're obviously available also to talk to you, and possibly if you're doing some research rotations in a lab, you'd like to come in and have one of your DNA samples sequenced in our facility here at UCI. Thank you.